My name is Deepak, and today my presentation is based on topoisomers. Topoisomers are very essential enzymes that take very important role in DNA replication and transcription. So, okay, um, now what we will do in the presentation are what is the topoisomers, what are the types, and what are the difference between the topomers, and what is the mechanism of that, and how why they are essential, and what are the inhibitors for them. Before going ahead, I want to introduce you some basic terminologies that are related to this presentation. First thing that you all know, DNA replication. This is the process in which DNA makes a copy of itself during cell division. The second thing is very important, is called supercoiling. Supercoiling is when the DNA undergoes additional twisting in the same direction or in the opposite direction from the turns in the original helix. It's the same thing that when it's like a telephone code, so it visible in this image when the supercoil when the helix is bound its own axis, then it's called a supercoil. There are two types of coils: positive supercoils and negative supercoils. Positive supercoils mean winding occur in the counterclockwise direction, and the negative supercoils mean winding occur in the clockwise direction. The third one is enzyme inhibitors. Enzyme inhibitors are that compound, yeah, agents that blocks the action of enzymes, yeah, interference. The fourth thing is linking number. Linking number is that how many times a strand binds around the helical axis. And the sixth thing is writing number. The number, and it means the how many times the axis of a DNA molecule crosses around itself in supercoiling. Okay. Topoisomers are the enzymes they change or alter the supercoiling of the DNA. So in the supercoiling, the supercoiling is simply I explain you that it it's a it's like a coil. So topoisomers cut transiently one or both strand. Uh, DNA supercoiling is very essential to DNA transcription and replication for the proper function of DNA enzymatic process. To topoisomers change value of L and W in the variation of W and reading number can change the state of DNA compaction in the cell. The other function of topoisomers are chromatin condensation, segregation of chromosome during mitosis and meiosis, recombination, and also DNA repair. Now we will talk about the topoisomers. There are two types of topoisomers. First one is type 1 and second one is type 2. The first one is work like it cut only one strand, one of the two strand of double stranded DNA. It relax and renew the strand. The second type is enzyme that cut both of the strand DNA helix simultaneously in order to manage DNA tangles and supercoils. The that discovered the James Wong is discovered the topoisomers one in 1971 and the Martin Gellert in 1976. Type 1 topoisomers always happen in eukaryotics, while the topoisomer second it, it occurs in both eukaryotics and prokaryotics. Type 1 topoisomers it does not require ATP hydrolysis, while the type 2 requires. There are three subclasses of type 1. Type 1 I topoisomers that called reverse gyrates. To type IB, second B topoisomers, and the third type is type C topoisomerase. There are two subclasses of topoisomer second. Type second A topoisomer that called gyrase. These gyrase the tetameric protein with two subunit A and B, and also the type B topoisomerase. Now we will talk about the mechanism of the type 1. In the mechanism of the type 1, this involves a formation of covalent DNA phosphatidylcysteine bond by nucleophilic attack of hydroxyl group on tyrosine to a phosphoric atom. It create a phosphodiester link between the enzyme. Also, DNA generate a free hydroxyl group. The another strand is placed in own place by binding non-covalently to enzyme. After the strand is resealed, by that it twists the DNA by one turn. By forming covalent enzyme DNA intermediate, the free energy of the cleaved phosphodiester bond is preserved, so no energy required for to reseal. For negative supercoiling, we use E. coli of type 1, and for both positive and negative supercoiling, we use eukaryotic type 1 can be used. In this image, the image, it's visible that how the three negative supercoils become two negative supercoils. First one is double helix. After that, it forms covalent DNA phosphodiester bond, and after it cut the strand and reseal DNA, and this way three negative supercoils become two negative supercoils. Now we talk about the working of type 2. In the mechanism of type 2 involves hydrolysis of ATP. In hydrolysis of the ATP activities of gyrase cause the conformational change. In the initially enzyme bind to one part of the DNA strand G stand G segment, G stand for gate, induce conformational change in A and B units. After a series of reaction occur in which G segment is cut by A and B domains of enzyme, and the end of the G DNA become a covalently linked to tyrosine. The ATP binding domains move towards each other, transport the T segment, their T stand for transport. Through the break, the G segment is resealed and the T segment is resealed by conformational change that separate A and B domain. The enzyme proceeds another. After that, the enzyme also proceeds another circle. Now we will talk about the inhibitors of topoisomers. There are many inhibitors. Why we need inhibitor? We need inhibitor because it stabilizes the intermediate cleavage complex, prevent DNA religation, and induce lethal DNA strand break. Novobikin or nalidic acid inhibited DNA gyrase. Novobikin blocks ATP binding, and nalidic acid blocks the breakage and rebinding of DNA. Also, there are many examples. In one of them, topoisomers 1 inhibit include irinotecan, topotecan, and captopricin. And also, topoisomer second inhibitors include ethopside, doxorubicin, and epiriboxine. Thank you very much.